Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, Ricardo Lopes, and today I'm here with Dr. Rob Dunn. He is the William Neal Reynolds Distinguished Professor in the Department of Applied Ecology at North Carolina State University. He is known for efforts to involve the public as citizen scientists in arthropod surveys and bacterial flora studies. His projects include studies of belly button biodiversity, mites that live on human faces, ants in backyards, and fungi and bacteria in houses. He is the author of 5 books, and today we will focus on his most recent one, Never Home Alone, From Microbes to Millipedes, Camel Crickets and Honeybees, the natural history of where we live, and Dr. Dunn just showed it to the camera, so thank you for that. And Dr. Dunn, thank you for taking the time to come on the show. It's a pleasure to have you on.
Oh, it's great to be on the show, Ricardo. I'm impressed that you said belly button biodiversity and didn't even smile. It was serious.
No, because since I have a little bit of a scientific background, my academic background is scientific. I guess that I take those kinds of things seriously. Yeah. Okay, so the book is very interesting indeed, and it has some very interesting insights that I really didn't think about before I read it. And the first thing I would like to ask you is, at a certain point, or in several different points even in the book, you refer to us as Homo indoorus, that is, the indoor human. So, you are trying here to make reference to the fact that at a certain point in our history, or even our evolutionary history, we moved from a natural environment to living inside houses, and that sort of altered the biological dynamics that we establish with other species. Is that correct?
Yeah, that's right. We don't know when the first houses were, but certainly 50,000 years ago, and maybe 200, 300,000 years ago, maybe before that. But as we build houses, we start to move into them, to sleep, to cook, and progressively over thousands of years, we started spending more and more time in them. And so in the U.S., the average kid born today, unless something dramatically changes, is going to spend about 23 hours out of 24 of his or her life indoors. And so I think that's a much more dramatic departure from our past of almost any other mammal, except for some really weird ones, than we've noted. And so, Homo indoorus is a little bit tongue-in-cheek, and yet I think that transition is a pretty serious one that we're not super cognizant of. And I've been reading a lot about futurists recently, and if you look at depictions of the future, they basically don't have an outdoors anymore. The future is all about the indoors as we're imagining it today. And so I think it's interesting to interrogate that idea and think about what does that mean about who we are. And in this book, I'm mostly thinking about what it means about who we're exposed to. And so if for the first 180 million years of mammal evolution, we were exposed to the species that, lived in trees, we're in the dirt, we're all around us. And now we're almost exclusively exposed to the stuff under our bed and in our kitchen. How do we think about that? And what does that mean for our potential futures?
Yeah. And this is interesting because during our evolutionary history, many people, when they think about humans, we tend to think of us as a particular living being that is separate from the rest, and that also happens for the other different species. But in fact, For us to be alive, we have to establish, ideally, some symbiotic relationships with other species, right? And also vice versa. So, in this sense, would you say that the fact that at a certain point we moved into houses and away from natural environments, let's say, at least directly exposed to them, that could also be changing our own evolution and the evolution of the species that live inside our homes?
Wow, that's a big question. So let me do it in parts.
Yeah.
Um, so I think there were sort of three parts there. One was really about identity. Does it change who we are? One is about does it change how we evolve? And a third is about does it change about how the species with us evolve?
Mm-hmm.
And if I take the first one, the identity one, I think it's very clear it's changed our identity. And I think one of the most conspicuous ways to think about that is to think about if you sit in a room with other people and you close your eyes, the dominant experience of those other people is their odor.
Mm-hmm.
And essentially all of our body odors are microbial. And so a reflection of the species around us and on us. And so to the extent that that's one measure of what it means to be you, as we've moved inside, the species on us and around us have changed. And so in some very real way, who you are is different than who you would be in some other context. And that might seem silly, but if we look at like skin microbes, there was a recent study of Yanomami hunter-gatherers, sort of small-scale agriculturalists in the Amazon, that found that one child in one Yanomami community had more kinds of bacteria on his skin than so far we've recorded from all North Americans.
Oh.
And so the question then becomes, is that Yanomami child's skin actually representative of what our skin used to look like in general? Have we changed that much? And we don't actually know. But it does position this possibility that we've already changed a lot due to those changes. The second piece is evolutionary, and so has our evolution changed? And that's trickier because the currency of evolution is reproduction and death, and so do we see different genes being favored now that we live indoors versus when we lived outdoors? I think for the most part, except for sexual selection and a few other aspects of modern human evolution, evolution's pretty dulled by the complexities of who we choose to date and what determines how many kids we have. That said, if we think back across the last 20,000 years, certainly as we moved indoors during that time period, there was the potential for really strong evolutionary changes. And so you think about, one of the first microbial changes as we moved into settlements and cities and indoor life in those places was a much greater exposure to fecal oral pathogens. Mm-hmm. And so it's easy to imagine in that transition strong evolutionary changes due to that indoor living. And there's one I can get back to. I'll try not to go too long here, but about armpits and how armpits have changed. And if that's of interest, we can revisit. Yeah. But then the other piece is, have other species changed? And that's the easiest one because, our lifespan is relatively long relative to our changes in how we live. Mm-hmm. The lifespan of the microbes on our bodies, the lifespan of the insects in our houses are really short relative to those changes. And so we've seen tons of rapid evolutionary changes in the species that live with us. And so in some ways, we're kind of this, we're stuck in a 20,000 or million-year-old body, depending on which feature you're looking at. And all the species around us are evolving quickly, and we're like with hands too big, can't grab at anything. Like, oh, why am I so slow? And so you can think about this in a stored food context, for example. One of the most common sets of beetles in houses is a grain beetle, a Psittophyllus species. that appears to have moved in with humans into grain storage areas in the Fertile Crescent probably 6,000 years ago, maybe more like 14,000 years ago. We don't know. When it did, it moved to eating our grain, but our grain is lower in nutrition than its ancestral food. And so it couldn't get enough nutrients. And so it evolved a special pocket in its gut that holds special bacteria that allow it to produce the vitamins it needs in order to persist on our crappy diets. And once it started to live with us, it didn't need wings anymore because we reliably carried it accidentally from place to place. And so it lost its ability to fly. And so there are lots of changes on that time horizon of these species moving in with us. And then the more rapid changes are things in response to our biocides. And so we spray pesticides around our house, and very, very quickly things evolutionarily respond. But I've talked for a while, so I'll stop there.
Yeah. So would it be rigorous to say that we have species living in our houses that are adapted to them, that is, to the kinds of materials that we use in building our houses, to the kinds of products that we have inside them, and even the ones that we use sometimes to try to get rid of them?
Yeah, that's entirely accurate. The tally of how many species that might be is a very open question, but the answer is many. And so you get some things like a German cockroach, which they enjoy Portugal like they enjoy the U.S. And German cockroaches actually can't live outside anymore. They only live indoors. So we think of them as indestructible. They can survive a nuclear war, but they can't survive our absence. And so there are lots of species like that. At the same time, there are species that are doing better in places like dishwashers that seem to be favored by the unusual conditions in dishwashers. And so we're seeing lots of species that are evolving to take advantage of the enormous new biome we're creating around ourselves.
Mm-hmm. And, I mean, would you say that we have both species that are nefarious to us and some others that are beneficial, both of them that evolved in our housing environments, let's say, or is it tilted more to one side or the other?
So if we consider, that's a good question. Beneficial for, I would introduce a third category, which is benign.
Okay.
So I think the vast majority of the species are benign. And so they're present, they don't do us harm, they're probably not beneficial, they're just there. You know, like most of your neighbors, right? Like they're not your friends, not your enemies, but they're present. And so that's tens of thousands of species go in that category. A tiny subset, and globally we're talking about, of common things like 20 to 50 are really dangerous, and we need to use every tool we have to keep them under control. And you know, and so this is stuff like, well, so for that subset of species, we need to keep doing the things we've been doing: wash your hands with soap and water, get vaccinated, all of these things. And then there's a subset of species that are clearly beneficial to us on our skin, in our bodies, around us. We tend not to know those species super well. And so there's a handful we're starting to understand. And this is like a great scientific failure, that how could we have neglected to study the species we need in order to survive? And yet, for reasons of history, we seem to have done that. And so what are the proportions? My guess is it's probably 90% benign, I got my percentages wrong. 9.99% beneficial, and 0.01, is that what's left over, percent super deadly. Something like that. And so if you're waving your hand around and thinking about what are the species in the air around me right now, mostly benign, some beneficial, and if you have a good public health system, the odds are that the really deadly aren't there.
So that's very interesting, because isn't it the case that until very recently, whenever people talked about these kinds of species like insects and bacteria and fungi living in houses, that they tended to think about them as always pathogenic and pests and some sort of germ, that is, every insect and bacteria and fungi we could have in our houses would potentially be a source of disease or would cause some sort of negative health consequences. But I mean, that's not really the best way to think about it, right?
Yeah, I don't know what the best analogy is. It's like you're in a crowded building and somebody tells you there's a thief in the building, and you just run after everybody you see. Right? The odds that you catch the thief are low, and the odds you accidentally tackle somebody who really is a nice person are high. And so I think when we initially started to figure out there were dangerous species in houses, which is very recently, we didn't understand germ theory, until 200 years ago.
Yeah.
The first actions were to try to figure out, well, what should we control? How do we control it? And when that started to happen, there was kind of a snowball effect of, well, if we can control these things, probably there's other stuff we don't know about yet that we should also control, which is it's not unreasonable. You figure out how cholera works. You figure out that cholera is killing people in just awful ways, and that you can control it by treating water systems, by disrupting the connection between feces and drinking water, by implementing hand washing procedures. And then you think, well, geez, what other stuff is in this water system? What else is in the house that could kill my kids? And so it's, I mean, absolutely reasonable to then think we need to hold this stuff back in general. And so it's hard to imagine a different history. It's hard to imagine a moment when we would have gone like, okay, we got cholera, but I bet the rest of them are good. But what happens, and the histories of this are not great, and my book is not really a history just of this component. But at some point, we forget the idea that some of these species, which we knew in the 1800s, we knew some of these species are beneficial or benign. We start to forget it, and then we get this feedback with the pharmaceutical, biocide, and antiperspirant and cleaner industries that were happy to reward our fear. And so as they started to do that, we suddenly became able to buy like anything we could imagine to keep our kids safe and to keep the dangers at bay. And so we ended up in this moment that I think is really extreme in the U.S. It's less extreme in much of Europe, but where we imagine, I mean, it is hard to find a building in the U.S. that doesn't have antimicrobial wipes sitting around, so that, you know, if you just want to wipe off what's sitting near you or like wipe the person next to you in a meeting, you can. And so we've really reached this really extreme point. And I think so it's a key moment to pause and consider, what future do we want? Is this reasonable? What should we be doing?
So, I mean, and has any sort of paradigm shift already occurred in how we think about the different kinds of species that live in our homes? I mean, in terms of going from considering probably most or all of them as pathogenic and pests to a point where we think that maybe we should be a little bit more careful about the ones that we should eliminate and the others that we should preserve, probably.
Yeah, so I think in the last 15 years for humans, we've been in the midst of maybe escalating is almost the right word, paradigm shift with regard to how we think about the species around us. and in our homes, but also more generally. And so just as an example of that, 6 years ago, if I would have given a talk and talked about a fecal transplant, which is to, take the healthy microbes of one person to give them to another, people would have laughed. Like, oh, what a joke. And now it's a medically very, very common procedure. And so a fecal transplant is really recognizing that there are healthy species we need and that they can go missing. And so that's very much part of this same paradigm shift about around us, on us, in us, our species we need. And so I think on some level that's changing and has changed very rapidly scientifically. What we're doing about that in our daily lives has maybe not changed as quickly.
Mm-hmm. Yes, that's very interesting. And I mean, you were also referring to what people call the biodiversity hypothesis, right? That is that because even more so recently, for example, children are less and less exposed to natural environments, probably during their development, they aren't in contact with certain types of microbes that would be beneficial to them, and also because even if people are not really aware of this, we have lots and lots of symbiotic microbes living inside us that really are essential for us to even properly function, right? And so, I mean, maybe what you were referring to was to also the fact that we have to take that into account and to consider the importance of biodiversity even for our own organisms.
Yeah, that's right. If we want to be healthy, we need these other species. And we need a plan for making sure they stay around.
Mm-hmm. Right. So, I mean, could you possibly give us some illustrative examples of species of, I don't know, insects or bacteria or fungi that are common in our homes that have beneficial effects?
Yeah, well, I can give two different kinds of examples. So one would be in our body. So Akkermansia is a gut microbe, just to make the extreme point. And nobody even knew it was common for a long time because most of the way people study gut microbes in your intestines, is to take fecal samples. And Akkermansia doesn't turn up in those samples very much because it turns out that it actually forms these tight connections with the villi and the gut and hangs out there. And so it doesn't wash out. But we now know if it's missing compounds, it causes the gut to be leaky and the immune system to do funny things in the gut. And so that's one really key one that even a decade ago, we basically thought was some obscure thing. We gave it an obscure name. And nobody thought to study it. And now it seems integral to daily function. And so that would be a gut example. I mean, another example would be if you open your refrigerator door, if you open your cabinet, I think we imagine that all the foods in those places are sterile until they go bad.
Yeah.
In reality, they're all full of trillions of microbes. And what you rely on is the microbes that don't cause you harm to sort of hold off the microbes that cause you harm until the food is expired. And so you're inadvertently relying on those microbes in there to do work. And so in your refrigerator, there are slow-growing microbes that live in each one of your foods that keep the other microbes at bay. If you buy a really stinky cheese, the stinkiest cheeses, these monastery cheeses, have on the outside a bacteria called Brevibacterium linens.
Yeah.
And it's a skin bacteria. It's a foot bacteria. Makes the cheese orange, makes it smell strong and taste delicious. But it also produces compounds that keep other bacteria from invading the cheese. And so at the same time, it offers great flavors. And it helps to preserve that cheese from other species. If you think about, your beer, your wine, your bread, your yogurt, The most common bacteria in commercial yogurt is Lactobacillus bulgaricus. Um, that is a strain of bacteria that comes from an individual Bulgarian dude. So Danone, who was an Italian yogurt maker, went to Bulgaria, gathered up a yogurt, thought it was really good, took it to Spain to sell it, changed his name to Dannon because it sounded, cooler for yogurt, and then made many, many more versions of this same microbe, which is a body microbe, and we now rely on to turn milk into delicious yogurt. And so these species are all around us, and they're very often very intimately associated with our bodies in ways that we don't imagine. That you can savor the flavor of a Bulgarian dude every morning, I think is something we don't think about. What would another example be? There are your skin microbes that help to defend you against pathogens. So you have a layer of Staphylococcus and Corynebacterium bacteria on your skin. If a pathogen lands on your skin, that's the first thing that the pathogen encounters. And most pathogens actually die before your immune system is even alerted because the bacteria on your skin kill those pathogens. And so I can go on. We're surrounded by good friends.
Yeah, and in terms of the insects, because insects, also because people can see them, are the ones that people, feel more disgust toward, I guess, but probably there are also some insects that could live in our homes and also could have some, could do something beneficial, even if it's only killing other kinds of insects, I don't know.
Yeah, so this is an interesting cultural one. So, I think our cultural perceptions of insects have changed a lot. And so around the world historically, it was common for people to favor spiders in their houses so as to help control other species. And so there's a Japanese poem by Issa, the Kobayashi Issa from the Edo period, and it's like, "Don't worry, spiders, I keep house easily," or messily. But with a recognition that spiders were providing a service. In both South Africa and Mexico, people historically brought in these giant webs of social spiders that would have thousands of spiders. They would bring them into their houses to control flies. Mm-hmm. The flies they were controlling very often had the potential to transmit dangerous bacteria. And so here was this, two things there. One is that spiders can be really beneficial to you, but the other is that the culture matters so much here that something that, I think for most of us today to imagine a social spider in the nursery with our kids, here's your baby and here are 1,000 spiders over your baby, is ridiculous. And yet different cultures around the world actually thought it was very reasonable. And so I think we need to be aware that we have a bunch of cultural biases toward these species, but to also note those spiders can help you. There's a spider in tropical Asia that specializes on eating the mosquitoes that transmit the dengue virus. Mm-hmm. And it prefers to eat them only after they're blood fed. And so when they're most dangerous to humans. And so it runs around the house, jumps on those spiders, eats them. If they smell a little bit like humans, the mosquitoes, jumps on the mosquitoes rather, it prefers them. And it looks like if you spray pesticides a lot, you kill off the spiders, but the mosquitoes still fly back in. And so there are these scenarios in which, we imagine, well, it has a benefit, but if I just got rid of everything, it would be okay. But we never get rid of everything. You're always trying to balance these systems. My other favorite one in this context is that there are a whole series of wasps that lay their eggs in the egg cases of cockroaches. Mm-hmm. So cockroach babies in most species come in these it looks like a little pill, and there look tons of little cockroach babies. Well, the wasps put their eggs inside the cockroach babies, the eggs hatch, and eat the living cockroaches from the inside out. Mm-hmm. And so if you don't like cockroaches, this is the best friend you could possibly have. And so there are these services.
Yeah. So we are focusing mostly on homes, that is where the places where people usually live. But you also refer in your book to places like hospitals and places basically where we receive medical care. And because people, since the development of the germ theory, are very worried about keeping medical places as sterile as possible. I mean, could that also be having some sort of negative health effects, at least in the long run, due again to the biodiversity hypothesis or not?
Yeah, yeah. I mean, I think that there are a couple of elements of the biodiversity hypothesis here. One is if you're missing biodiversity, your immune system starts to do bad things. It essentially gets bored and attacks whatever it finds. And so for sure that can happen in hospitals. It happens if we think about C-section babies. C-section babies don't. If you're born vaginally, the microbes you get when you're born mostly come from your mom. And so they're gut and vaginal microbes from your mom that establish in your gut. And this is what mammals have done for hundreds of millions of years. If you're born C-section, the most likely microbes that you get in your gut are hospital microbes. And so one part of this hospital story that is that in as much as our lives start in hospitals, hospitals are dramatically changing what our normal species that we carry with us are. And so that's one part. Yeah. There are now big studies showing lifelong effects of whether you were vaginally born or born via C-section. But the other thing is, think about just going into a hospital in general, which microbes are present. Well, in a surgical environment, you can actually sterilize a surgical environment totally for a very brief window. And that's more or less what you want. You want those environments. When you open the body cavity, when you open a wound, you want those environments to be sterile, if we can do it.
Sure.
But the rest of the hospital is not sterile, not even close. So long as people are walking through it, so long as there are walls and floors, it's full of microbes. And the microbes that we tend to favor in those environments are ones that do really well despite being sprayed with antimicrobials, doused with bleach, doused with antibiotics. And so we have the potential in hospitals to create environments where the bacteria that are hardest to kill have a chance to do really well. Now, the good news is that most of those bacteria, if you have other bacteria around, they're wimps. They're not very competitive, in part because the genes you need to be resistant to many antibiotics, it's like a big, what's the best analogy? They're just really costly. And so it's like you can have the tools to compete, or you can have the tools to be resistant to antibiotics. And you can't have them both. They're very hard to have them both. And so if other bacteria are present, those resistant bacteria tend to do very, very poorly. And so there's the potential to manage that interaction. So we're disfavoring those really deadly things, but we tend not to do it. But I think we have the tools to imagine very different futures in that regard. And the other thing, oddly, that's like a hospital are industrial animal production facilities. And so chicken farms, pig farms, hospitals, very, very similar to each other microbially, in that we're favoring really unusual microbes that can't live anywhere else.
Yeah, that's very interesting. Let me ask you now, so we've already referred to, for example, bacteria and insects that we have in our homes. But I mean, another very common type of species that we have, or I mean, two types of species are cats and dogs that are probably the most common pets that people have at home. So, I mean, in terms of them, are they mostly beneficial, or do they also, or can they also bring some sort of negative consequences for our health, and how?
Um, you ask these giant questions, Ricardo, and then I talk for too long. So I'll try to be succinct. It looks like in urban contexts, dogs seem to be largely beneficial. And the reason they seem to be beneficial is that when we fail to be exposed to the microbes we need for our immune systems to work properly, dogs seem to be this kind of last connection to nature. They bring stuff in on their feet and their fur. And as a result, families who have a dog when the mother is pregnant, the child when born is much less likely to have allergy and asthma in cities. It seems much less pronounced in rural regions, which is what you would expect if those kids are already getting the exposures they need. We also know the single biggest predictor of which microbes are in a house is whether or not you have a dog. And we can actually tell if you have a dog based on what microbes you have, which is kind of a crappy party trick because you already know if you have a dog, right? My guess is that as we understand more, in general, the more we've lost a connection with nature, the more that any mammal that we let in and out of the house is going to have some beneficial immune effect. But we mostly know it for dogs. We can't rule it out for cats. That said, so far nobody's shown a benefit of having cats. I mean, probably they make some people happy, but immunologically nobody's shown a strong benefit yet. And we do know that cats can bring in parasites that have really surprising effects on our biology. So do you have a cat, Ricardo?
No, I already had one, but at the moment, no.
So when you had a cat, right? So that one of the parasites that can infect cats is one called Toxoplasma gondii, and 20 years ago, people mostly heard about this in the context of pregnant women. That parasite can infect pregnant women, it can infect the fetus, and it can cause really devastating effects for the mother and fetus. But that was mostly how people heard about it. And slowly what emerged was that in nature, the other thing this parasite did was that for its life cycle, it has to get into cats, and it's only in cats that it can have sex. But its first host is very often rats and mice. And so the question for a while was, how does that work? And it's now pretty clear that the parasite, when it gets into rats and mice, it gets into their brain, it produces the precursor to dopamine, and it makes them act more risky, and it makes them actually no longer afraid of the smell of cat pee. And so it looks like the parasite's actually manipulating the brain of the rats and mice to make them more likely to be eaten by a cat so it can fulfill its hopes and dreams. And so this has been known for quite a while in mice and rats, and nobody cared about it at all because it was like, oh, look at that weird thing nature does. Yeah, nice job, ecologists. And more recently, it's become clear that humans who are infected by this parasite also seem to show altered behaviors. And so there's evidence that if you're infected by this parasite, you have you tend to be more risky, you show a changed personality profile. It's one of the predictors of the onset of schizophrenia. And so here's all this stuff that's happening because of this strange parasite that evolved in the context of predators and rodents in the wild. And in and of itself, and this one's in the book at some length, but it's interesting. But I also think it's an indication this happens to be one that we've studied well. Yeah. What are all the other things that we're bringing in with cats and mice and dogs and ferrets and guinea pigs? And I mean, we have a bunch of people in our study that had indoor goats. what will we know in 30 years about these species that we don't know today? And my guess is there are huge surprises that we're being affected by species we don't even know exist at the moment.
Hmm. And also probably learning more about the effects that other less common pets might have in our health and in our houses, right?
Yeah, right. Less common pets, less common foods, all of the things we bring in with us, um, and no one's ever really studied. I mean, a simple one would be, which microbes are you exposed to in your salt shaker?
Mm. Yeah.
We have no idea, and yet we shake it on every day.
Yeah.
And we know there are microbes in there. There are actually microbes that live inside the salt crystals,
Yeah.
and they're actually square shaped so as to maximize space use. And they're there. If you make a washed rind cheese that has salt on the outside, some of those microbes become common. They're actually marine microbes on the outside of those cheeses that give them some of the flavors. But what effect do they have on you each day you shake that salt on?
Yeah.
Check back in 20 years.
Yeah, and since you're referring to food, I mean, because nowadays we have more and more, or we have food that is more and more processed and goes through several different steps in its production, and I mean, it's probably also altering its microbial composition. I bet that it's not sterile. It's not sterile at all, but I mean, we are probably modifying in unexpected ways its microbial component. So, I mean, do we know anything about that? Or
There's some suggestion in the literature that I mean, think about fruit. That your exposure to microbes in fruit has dramatically changed because of the way we grow fruit now, because of the way we clean fruit, because of fungicides, pesticides. And so imagine, I mean, if to take the case example, one fruit that's being gathered by a chimpanzee, our closest living relative, and one fruit that you're getting from a store with a waxy coating on the outside. The microbes in those two they're not even comparable. And so that's a very different effect. And now what does that do to your gut? Hard to say. There is a book that just came out this week, today maybe, deals with in general how do fermented foods affect which microbes are in your gut. And so it's a first attempt to try to figure out, like, if you eat lots of cheeses with live microbes, kefirs, name your yogurts, how much does that change what's in your gut and your ability to digest other things? And so I think that will be a growing field. And in some ways, that's the extreme case of your question, because there are microbes on the outside of the apple, but the biomass of the living mass of those microbes is not so great. But if you take an apple-sized dose of yogurt, the living mass of those microbes is extraordinary. And so probably the effect of the fermented food is much greater. And it's worth remembering that that's a really old story. I mean, John Spaeth at the University of Michigan has now argued that Neanderthals may have fermented meat 200,000 years ago. And so the odds that we were getting huge doses of microbes in our guts from our food for thousands and thousands of years, pretty high. And so that's a big change. The trick with a lot of this stuff that's at the intersection of our immune system, our gut, our brain, and food is that you sort of are at the intersection of four of the things in the living world that we understand most poorly and are most complex. And so my tendency is to be suspicious of any claim about that intersection that seems super clear. That, like, we figured it out. Anybody right now who says we figured it out, the odds that they're full of it, really high. Because we're still so early in this story. We're in this dark cave and we've got a candle that keeps blowing it out, and we're lighting it with stones. And so, as a scientist, that's a beautiful thing because we got work to do. But as somebody who then goes home and tries to live my life and figure out how do I be healthy, how do I take care of my kids, it's extraordinarily frustrating because I think we think of ourselves as the fancies. We can recognize faces using computer algorithms. We can make transgenic babies, and yet we're so ignorant still. And that will continue to be one of our great struggles.
Mm-hmm. Yeah, let me ask you now, so you've already gave some examples of that, like for example, you referred to a type of bacteria that tends to develop in our dishwashers at home. Could you give us more examples of parts of our houses or pieces of technology or even materials that we use usually in our homes where we are favoring the development of, for example, microbes that might have some negative health effects?
Early days. It's early days of that study, but I would say that there seems to be a sense that in environments where we really create hard conditions, where it's hot and then cold, wet and then dry, acidic and then basic, that in those conditions we're very often favoring microbes that are able to withstand all of those things. And if they can withstand all of those things, we're increasing the odds that they can also live on us. And we don't understand this super well, but it seems like this is something to be cautious of moving forward. And so, dishwasher dish would be an example. The dishwasher's hot then cold, it's wet then dry.
Yeah.
It's warmer than the human body. This is for a lot of microbes, our bodies are too warm to do well, especially fungi. And so fungi mostly don't live on our bodies. There are very, very few exceptions. If you're immunocompromised, that's different. But for the most part, they struggle. But if we repeatedly expose fungi to really hot conditions, it ups the odds that the lineages, that the genes that allow them to do well in those conditions will be favored. And so there are some folks who've raised some alarm about this. Are we inadvertently favoring those kinds of microbes? Another weird example, but a pretty important one, is that one of the fungi, for as much as many of the bacteria in houses are beneficial or benign, a subset of the fungi are clearly problematic. And so one of those fungi is Stachybotrys chartarum, which is the toxic black mold. And we don't understand it super well. It's a pretty mysterious fungus. And one of the mysteries for a long time was how it was getting into houses, because it has great big spores, doesn't seem to fly through the air. We sampled air a lot in houses, we don't see it. Birgitte Andersen in Denmark has recently shown that at least in Europe, and I strongly suspect also in the US and elsewhere, when you brand new drywall, the drywall, is that what term would you use for it? The wallboard, so composite white wallboard, that Stachybotrys is already in there preloaded. And so if that drywall gets wet, Stachybotrys is like, it's the happiest fungus in the world. It's just waiting there. And so if your house never gets wet, Stachybotrys is almost certainly never going to be a problem. But if your house floods, and in the coming years we expect lots of floods, it is just already in the house waiting. Yeah. And so that's the kind of thing, it's easy to fix, but we have to know it's happening. And so there's some stuff like that. Another example that's a tricky one is in shower heads, that non-tuberculous mycobacteria are becoming more common. And in most people, they're not a problem. But if you're immunocompromised, they're a problem. And a lot of people are. And it looks like one of the things that's favoring these bacteria is in places where the water that comes from aquifers needs to be treated because we polluted it, or needs to be treated because there are no natural aquifers, and so the water has to come from impoundments. In the U.S. especially, and also in Europe, we often chlorinate that water. And that chlorination disfavors a lot of the sort of ordinary microbes that are harmless, and it leaves the microbes that are chlorine tolerant. And it looks like non-tuberculous microbacteria are pretty chlorine tolerant. And so seem to be inadvertently favoring them. That problem is a real jerk in that if your water system needs to be treated, you have to treat it, right? It's way worse to have a water system in which you get cholera building up or in other problematic microbes than it is to have this bacteria. And so the trick is that if you have an aquifer that is safe and healthy, not to screw it up. But once you screw it up, we only have so many options of how to replicate that in a way that provides safe drinking water. And so, and I think buried in this is the lesson that when we have functioning ecosystems we rely on, we have to save them because we're too stupid to rebuild them from scratch. And so that's kind of an elaborate example, but it gives you a sense that often these stories are very complex. And so now scale back and imagine in your house are tens of thousands of species. Most of them nobody's ever studied, but each one has a story as detailed as that non-tuberculous mycobacteria story. And so it's going to take a while to pull all these pieces apart.
Yeah. So let me just ask you one last question, and this is one of the solutions that I guess we could say you present in your book to try to solve the problem of loss of biodiversity in our homes, hospitals, etc. You talk about gardening the microorganisms that we need. So could you tell us what that is about and how we should go about it?
Yeah, I guess two quick things there. One is that if you live close enough to a place with wild biodiversity where you can get your hands in the dirt, you can get your kids outside, that's the best solution, is to not become Homo indoorus, like hands down. But if we're going to become Homo indoorus, if we're going to spend our lives indoors, we need some way of making sure we have the species we need to survive. Mm-hmm. And so on some level, that's going to require some kind of gardening. Mm-hmm. The trick is we don't have a great understanding of what we need to garden. That's not going to stop people from selling you things. And so there are already products in the market in Europe, for example, that you can, they're microbes you spray around your house to benefit yourself. Some of those are probably beneficial. They're not super well studied. But the place when I was finishing the book, I was looking desperately for people who would have a good gardening solution. How do we garden the future? And I thought architects would give answers, and I didn't find the architect with the magic. Maybe they're out there. If you're out there, contact me. But where I did find it was in bakers and people who are fermenting in their kitchens. And for me, one of the really beautiful stories was to work with people who make sourdough bread. And so to make sourdough bread, you mix flour and water. It favors bacteria and yeast. The yeast produce carbon dioxide. The bacteria produce acid, which is what makes it sour. Yeah. And we benefit from those. They make delicious bread. It's wonderful. We should be grateful every day for their magical microbial powers. But those bacteria that make the starter sour also kill other bacteria. Mm-hmm. And so the starter is always safe if it's acidic because the other bacteria have been killed off. And so on the one hand, making sourdough bread is just good, harmless hobby that is tasty and delicious. But on the other hand, I think that's a metaphor for what we might imagine moving forward. Can we grow gardens that weed themselves? What would that look like? And in the meantime, while we're thinking about how to do something like that, in many of these cases, moderation is better than what we're doing now. So you don't need to rethink your life entirely. Walk outside more. Get your hands in the soil more. Leave your windows open more unless you live in Beijing. Um, a lot of what would be better is simple. Use soap and water. Don't use antimicrobial wipes. In the short term, those are all very reasonable things you can do today. In the long term, though, if you're imagining the future, if you're thinking about what would it look like to live on Mars, what should the homes look like in 2050, include the garden in that future. Include that in our thinking about how do we imagine what we should be doing. Because to fail to include that garden in our houses is to assume either that we can kill everything, which is a nightmare, or it's to leave it to chance, which also seems stupid.
Mm-hmm. But, I mean, if we are gardening certain types of species, we have to know more or less well what we want to garden, right?
Yeah.
Mm-hmm. So we have to know probably what we should include in our garden and the sort of conditions that we would expose that garden to, correct?
Yeah, and I think that requires radical re-envisioning of how we think about microbes in our daily life and how we study them. If we think of our first gardens, the dawn of agriculture 14,000 years ago, it was hard. And it created a new kind of society. But it required radical new innovations, and it had problems. If we're going to garden favorable life in our houses, it's also going to be hard, and it's going to require major work to figure out how to do it well. It's not a toy problem. It's a big problem. And so on the one hand, in some ways, it seems really daunting to me to think about how we do it. On the other hand, I know that every day I'm in a building, I'm in a world where we're inadvertently gardening scary shit. And so is a beautiful garden more scary, more daunting than our existing one, which is a garden of demons and unusual dishwasher fungi? I don't know.
Yeah, okay, so let's leave that question open for other people to answer in the future, hopefully. And Dr. Dunn, I would just like to ask you to please show the book again to the camera. Again, Never Home Alone, From Microbes to Millipedes, Camel Crickets and Honeybees, The Natural History of Where We Live. So I will be leaving a link to the book in the description box of the interview. And just another thing before we go, Dr. Dunn, could you tell people what are some of the best places on the internet for them to find your work?
At the robdunnlab.com, you'll see a bunch of my work. That's probably the most reasonable place. That's where we gather a lot of it. But my books are all over the place too, and here and there. And there aren't that many Rob Dunns. And you can Google me, and if you find a different Rob Dunn, maybe he's got something interesting to say too.
Okay. So I will be leaving some links to your work and also to your books in the description box of the interview. And Dr. Dunn, again, thank you a lot for taking the time to come on the show, and I really love the conversation.
Thanks so much, Ricardo. Thanks for doing what you do. I think it's a great service.
Okay, thank you. Hi everybody, thank you for watching this interview until the end. As you might have noticed, I've started this channel in February 2018 and have been putting out regular interviews with academics and intellectuals from a variety of fields. To keep the channel sustainable, I would like to ask you to please visit my Patreon page and to consider making a pledge there. Any amount, even just $1, would already be a great help. There's also links to PayPal in the description box, box of the interview. Otherwise, and if you like what I'm doing, please share it, leave a like, and hit the subscription button. I would also like to give a huge thank you to my patrons and PayPal supporters: Karen Litska, Anne Blanchett, Perel Galarson, Lau Guerrero, Chantel Gelinas, Francis Ford, Hans Frederick Sunda, Brian Rivera, Sergio Condriano, Janne Henninen, Ricardo Vladimiro, Craig Healy, Adam Castle, Vega Guidi, Olaf Alex, Jonathan Wiesel, David Diaz, Anian Kata, Jacob Klinkby, Matthew Whittingberg, Drs. Jerry Mueller, Herbert Gintis, Rutger Voss, and Bo Weingart. My three producers, Isar Weber, Rosie, and Jim Frank, and my executive producer, Michal Ruzieski. Thank you for all.